All right, let's continue on. So we just took a look at this slide in the previous mini lecture and talked about the difference in the terms DNA, chromosomes, and chromatin. So I think most of us, when we think about eukaryotic DNA or eukaryotic chromosomes, when we hear those words, we remember seeing pictures that look like this, these kind of X-like structures. And they are, floating around here in this ball that is the nucleus. And if we look here and here, that's a pair. So this could be chromosome 1, chromosome 2, all over here it's telling us it's chromosome 6, and so on. Over here, those pictures have been laid out by size, which correlates with the numbering of eukaryotic chromosomes. So the largest chromosomes are chromosome 1 in the human genome. And down here we can see the XY pair. So the presence of the Y chromosome typically imparts maleness, at least in humans, not necessarily in all eukaryotic genomes. And to lay the chromosomes out and image them this way is to make a picture that is referred to as a karyotype, K-A-R-Y-O-T-Y-P-E. This is a karyotype. And that's the job of a cytologist. So anytime that a woman has a pap smear done, that they're making a karyotype or imaging the chromosomes that are present in cervical cells to look for abnormalities that might indicate or suggest cervical cancer or the risk of cervical cancer. So the thing is that these kinds of pictures can only be generated during certain times of the cell's cycle. And so for the vast majority of time, you won't see structures like this when you look at a nucleus. So you'll actually only see these structures during prophase of mitosis or prophase of meiosis if this was a gamete-forming cell. In the somatic cells, it would always just be prophase of mitosis. So these structures occur after DNA replication and during prophase when their chromosomes are getting ready to divide. So most of the time, you'll have cells that are in interphase. And in that case, the DNA molecules are complexed with protein and RNA in a spaghetti-like ball that is referred to as chromatin. So chromosomes are in fact only visible at some points in the cell cycle. And here's an actual image of some plant cells, and we can see here's a nucleus that contains DNA as found in the structure called chromatin versus DNA that is organized into chromosomes, these really thick strands that the cell can then literally get a hold of with the cytoskeleton and pull apart to the poles of the cell during and in allowing mitosis. So this image gives us, it's just a drawing, it's a cartoon, but this is a little bit more really how the DNA would typically be found in the nucleus of a eukaryotic cell. And it would be found in this structure called chromatin. And one of the reasons for that, or one of the accomplishments of chromatin, is that it's part of how we're able to fit pretty giant genomes into pretty tiny spaces. So we're going to actually compact or pack our DNA molecules down 10,000 times. We're going to make them 10,000 times smaller through organizing them in these structures called chromatin. So what is chromatin and what does it do? It's a term that is used to describe the complex of DNA, protein, and RNA that is found in all eukaryotic nuclear genomes. And what it accomplishes is shown here in 1 through 5. It helps to package DNA, it helps to compact it, I just mentioned that. It helps to protect DNA from damage, for example, from oxidative stress or even physical damage. It facilitates the processes of mitosis and meiosis, both of which require genome duplication. It helps in the regulation of eukaryotic gene expression. And then 5 is a little bit of a mistake here, so this is just sort of an extra note that I'll get to. But it's not what chromatin does, but how it's organized. So really it's 1 through 4 are the what chromatin does, and this is sort of an extra thing about the structure. And that is that chromatin can be found in 2 separate forms. It can be euchromatin or it can be heterochromatin. So let's think about that next. This is an actual electron micrograph, and it is showing us the nucleolus, which I mentioned in the previous mini lecture. And we can see the nuclear membrane bounding this nuclear structure. These are various organelles out there. This is actually the endoplasmic reticulum all around here. And this light material, which is called euchromatin, and the darker material is called heterochromatin. And so these are just largely defined based on how they look when we're studying them through a microscope, but they also have, these two states have functional significance because the euchromatin will contain active genes, and active means regularly used to make functional RNA or protein, more specifically peptides. Whereas the heterochromatin is going to contain inactive genes, ones that have been turned off, for example, the second X chromosome in women, something we'll talk about in future, and also repetitive DNA sequences, like the sequences that are found in telomeres. All the telomeres are like tucked inside these little areas that are referred to as heterochromatin. So I mentioned a minute ago that the heterochromatin, which stains darkly, tends to not have active gene encoding sequences within it, has a lot of repetitive DNA, which means you see the same sequences over and over and over again, like AAA, 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 and so on. And one type of repetitive DNA worth mentioning is satellite DNA. So satellite DNA means you have a sequence that is 5 to 300 base pairs long, and that depends on the species, and it repeats over and over and over again, and I mean up to 1,000,000 times. Okay, and that stuff is not coding. So it serves other roles in the cell, and what those roles are, we're not entirely sure, but they seem to be roles that contribute to the complexity of the overall organism. That kind of DNA, this stuff called satellite DNA, is generally found as heterochromatin. So back to chromatin. Chromatin contains proteins, right? It contains DNA, it contains some RNA, and it contains proteins. So the kinds of proteins that are found in chromatin, two important categories are the histones and the non-histones. So we're going to talk about histones H1, H2A, H2B, H3, and H4. Now, all histone proteins are small and they're basic, which means they have a positive charge. And why is that? It's because they're rich in the two amino acids lysine and arginine, which are small, positively charged, or not small, but they are positively charged amino acids. The proteins are small because there just aren't that many amino acids in them. Now, you should remember from our discussions about gel electrophoresis that DNA, due to its many phosphate groups, has a net negative charge. So these small basic proteins with their positive charge are able to attract and hold DNA because positive and negative attract. So what? Okay, remember that rule for eukaryotic chromatin, that it was important in allowing it to compact. Well, how much? So it's kind of like this big cat trying to fit itself into this little tiny basket, which is something that my cat and cats have always loved to do, is to try to wedge their big bodies into little tiny spaces. It seems to be kind of a cat thing, but similarly, a challenge for eukaryotic cells is to wedge all kinds of DNA into a little tiny space. So in humans, for example, our chromosomes range in length, or our DNA molecules more accurately, range in length from 16,000 to 85,000 micrometers, chromosome 1 being the largest. The nucleus itself is only 6 to 10 micrometers in diameter. So we have to pack all this material, on average, 10,000 times smaller in order to fit it into the nucleus. And how do we do that? We do that by organizing it into chromatin. And the first stage in organizing DNA into the structure called chromatin involves the histone proteins I mentioned a moment ago. So the histones are going to organize into a structure that is going to be called a nucleosome. And this is what a nucleosome core, or histone core, looks like. It's got two layers, like a birthday cake. And the top layer is divided into 4, and the bottom layer is divided into 4. And they repeat. So the top layer is identical to the bottom layer, and then it has an H4, an H3, an H2B, and an H2A. And the bottom layer is just like that as well. So what? Remember, these guys are positive. So the negative DNA, which is shown here as light blue, is going to be very attracted to it. And so DNA as a strand, as a double helix, will wrap, and there's the double helix, you can see it in there, will wrap itself around a certain, like not quite 2 times, but like 1.5 times around, and it will form this structure. And these structures look like beads on a string, but each of these is a nucleosome. So they look like beads on a string, and they're called nucleosomes. And this little protein here is the H1 protein. It's a histone protein as well, and it helps to pull the nucleosomes all together so that we're condensing DNA. So instead of having it all stretched out, we're wrapping it around these histone cores, and then we're pulling them together with another histone, and that's the start of making chromatin. So over here in this cartoon, there's the double helix, and then we're wrapping the double helix around histone cores, which are shown in green here and in shiny purple here. They look like Christmas balls, Christmas tree balls. We're wrapping that DNA around, and then we're using histone protein to help pull them together. And then this structure will be further compacted with the help of non-histone proteins into a larger string called a solenoid. And that is what makes these spaghetti-like structures that are referred to as chromatin inside the nucleus. And that concludes this set of 2 mini lectures that address general properties of eukaryotic genomes and chromosomes.